All right. Well, welcome everybody. Welcome to our 12th iteration now of like the CMFI mass spec seminar. Yes, it's my great pleasure to have Professor Hannes Link give the lecture today. I'm very much looking forward to learn more about how Hannes and his lab uses flow injection analysis with mass spectrometry to get like metabolomics profiles really fast and at scale. So many of those. But yeah, before Hannes gets started, just a couple of words. So Hannes is a biotechnologist. I think he got his PhD from like the Technical University Munich, before he then moved on to do a postdoctoral research stay at the ETH Zurich in Uwe Sauer's lab, before he then became like an independent group leader with like support from an Emmy Noether Fellowship in Marburg at the Max Planck Institute. And then I think the SIMPHI was quite lucky to convince him to then start here as a W3 professor 2 years ago, 1 year ago. Time runs.
Time was two years.
So yeah, I have the pleasure to kind of like sit in the office next to him, and I guess most of you know him too. So yeah, without further ado, thanks a lot, Hannes, for doing this, and yeah, looking forward to your lecture.
Great. Yeah, thanks, Daniel. Also, thanks for inviting me to this really nice series where you give an overview of all the metabolomics methods. And I just share my screen. Hope you can see that. So now you should see the presentation in the right mode. Is it in presenter mode? Daniel? Yeah. It's good. Okay, great.
That's good.
Yeah, so as Daniel said, today I want to show you what we do with flow injection mass spectrometry, how we do metabolomics with these methods. And it's really the most simple thing, I guess, you can do with a mass spectrometer. You just record MS1 spectra. There's no column involved, nothing. So really very basic. And I hope I can show you today a bit what it requires for data processing and what you can do with this method. So the outline is a little introduction about flow injection, and then I will spend some time, as I said, on data processing, what we do with these data. And then I will show you a little stress test that we made. So we tested how good is this method actually, what can it do, and what are the drawbacks. And at the end, there's a case study from the lab where you had a CRISPR screen that we did with flow injection, and we actually put some of the data on the GNPS dashboard, and yeah, then you can have some hands-on experience with VR data. So as an introduction, just very briefly, what my group is doing so far, we worked mainly in two areas, systems biology and metabolic engineering. So in the systems biology area, we are interested in interactions of metabolites with the expression of certain genes. So these metabolic genetic networks, how we can detect them, what is their function. These are these studies. And then we kind of use this knowledge to engineer better strains for biotechnological applications. So that's metabolic engineering. And since we are in Tübingen, we moved now into some newer fields for us. It's microbiome engineering and also investigating interactions of metabolisms and drugs. And in all of these fields, it's very important for us to measure metabolism and also to measure metabolism in a very fast way, because one of our main approaches is a multi-omics approach, where we use a genomic approach. So we create many strain variants. So we use mainly CRISPR, where we have hundreds, thousands of strains. And then we combine this with our metabolomics methods to see what is happening in these cells. And the data we get looks like this. So we have many metabolites measured in many strains. And then to this data, we apply some computational analysis to infer networks, or at the end, eventually to arrive at testable hypotheses. And you can imagine if we have many of such strains, what we need are fast metabolomics methods. Maybe for hundreds of strains, it's still fine to work with the LC columns. But once you move in the thousands of strains, you really need fast methods. And so this slide is just to show you that we did this in the past, mainly also with LC columns. So what we did is we used these very short Hillig columns to measure polar metabolites. These are 3 centimeter long Hillig columns. We use a 3-minute gradient. So in total, it's around 3-minute runtime. This is already pretty fast, but the drawback here is we use a targeted method on a triple quadrupole mass spectrometer. So in this limited amount of time, we're also limited with the scan rate. So we can measure around 50 metabolites in 3 minutes. So this is the maximum we got from this fast analysis. And if we want to go for more metabolites, higher coverage, and even faster analysis time in the 10 to 30 second range, that's where we use flow injection mass spectrometry. And as I said, it's really simple. So there's no column involved. So we directly inject the sample into the mobile phase. It's a simple mobile phase, which enters then a high resolution mass spectrometer. And we learned it works best with a time of flight mass spectrometer. We also tested this with the Orbitrap, but there you have some other problems. So the best thing is to do this on a time of flight mass spectrometer. I will show you later why this is exactly the case. And then you can do different things with these methods. So as I said, one of our main applications are CRISPR screens in E. coli. So at the moment, we're working mainly with 2 libraries. So one is a library with CRISPR interference, where we can knock down certain gene. It's indicated here. We use a deactivated Cas9, and then we can just block transcription of a certain gene. There we work with around 7,000 E. coli strains, where we target different genes. And then we have another CRISPR library. This is now with CRISPR genome editing, where we inserted point mutations in all essential genes of E. coli. And these are more than 50,000 strains, which Torben Schramm from my group has developed. Another application is, this is back from my postdoc time at ETH Zurich in the group of Nicola Zamboni and Uwe Sauer. There we just used flow injection mass spectrometry to do something like real-time metabolomics. And what we did is shown here. So in this picture, you see a setup where we have E. coli culture here, and then we pumped this E. coli culture directly into the mass spectrometer. So without extraction, really the live cells, they enter via this setup of 2 6-port valves. They directly enter the electrospray. There we applied a high voltage and high temperatures, and then we could really monitor what is happening in these live cells. And in this paper that is cited down there, we did the following experiments where these cells starved for carbons for 2 hours. So for 2 hours, we injected every 5 minutes a sample here into the mass spectrometer. And then comes the interesting phase where we then looked how they exit from this starvation. So we increased the sampling rate. So every 15 seconds, we injected a sample into the mass spec, and then we followed for 30 minutes this exit of starvation phase. And the data we get is indicated here. I will not go into the details. All that you see is here over time. We see then how the different ions that we can annotate to metabolites are changing. So some are increasing, some are decreasing, and a lot of interesting dynamics. But as then, I will not go into these details. Rather show you now a bit the pros and cons of flow injection mass spectrometry and how we actually work with the data. And the data looks like this. So it's very simple. Just one MS1 spectrum per sample. So we do this over a mass range of 50 up to 1,000 or up to 1,700 sometimes. And then we just have the ion counts here on the y-axis. And the advantages, of course, it's very fast, 10 seconds. At the moment, we use 30 seconds per sample. Another advantage, there's no need for a specific LC column. So we can capture polar compounds, but also non-polar compounds with this, and we don't have to adapt a specific LC column or any gradient. And also the data analysis, as we will see in a minute, is very simple because we're working with 2-dimensional data. Of course, there are disadvantages. So the selectivity or the specificity is very low. We only have MS1 level information, so we are never certain if it's really the metabolite. So we lack the MS2 level information and also the level, the information that we get from the retention time. And then also something we learned and we tested now extensively is the effect of the matrix and what happens in the electrospray. Because if all analytes and the matrix, they enter the electrospray at the same time. So you can imagine this method is very prone to matrix effects and also to ion suppression. And of course, everything with the same mass is impossible, not impossible. So there are ways to distinguish. It's very difficult to distinguish isobaric compounds, isomers. So I will show you now in the next couple of slides what we're doing with the data. So as you've seen on the previous slide, so what we get from the machine are these .d files, which contain these MS1 spectra. So these is from the Agilent machine, the .d files, we convert them with MS Convert into MZ XML files. So they're simple file conversions. So we apply no filters, no centroiding, because the centroiding and the spectral alignment, this happens then in some in-house software with MATLAB. So the first thing that we do is, here you see the scans over one injection. So one simple peak, this is the 13 seconds. So we get around 35, a bit more than 35 scans. And then we take the top 10 scans where we have the highest total ion count. So they're indicated here as these little dots. And from these top 10 scans, we then just average the spectra and get something like this. So this is the average spectrum per sample that you just saw. And we do this for many samples. Now, the important thing what we do here is also that we resample these spectra to ensure that all of our samples have the same M to Z vector. So we kind of align them, and that allows us to get a data structure that looks like this. So as I said, we have these many samples. These are the rows in this data structure. And then we have in the columns the different M to Z values. And because we aligned them, we have the same M to Z vector for all our samples. And then we get data matrix that looks like this, where we have all the intensities, right? And if you would now get one of these rows, this would be one of the spectrum from 50 up to a mass to charge ratio of 1,700. Then comes the next step. So we're merging all of these spectra. That's what you see here. And we do this in little windows of 100 millidalton. So this is again the matrix that you just seen on the previous slide, where we have the many samples here. In this case, it's a data set of 252 samples. And then we just merge them all together. And each line here in the spectrum is one of the rows here in this matrix. The next step is then we do the centroiding and the annotation. So what happens then? First step would be we pick all the peaks that we can find in this window. So peak picking would be everything with apply certain cutoffs. So everything with intensity higher than 1,000 ion counts and a prominence higher than 1,000 ion counts we pick. So these are all the little dots in color that you see here, even this little dot here very near the baseline that you see here at the bottom. So this is the peak picking. And next step, we try to cluster all the peaks that seem to have a common mass to charge ratio. So you see this here, so everything which is close to each other, we cluster together, and then we just assign the average mass to charge ratio in that cluster. Here in the left, you can see it a little bit better. So there's some variation in the mass to charge ratio of the individual samples, but we cluster them together. And then the dashed line, this is the common mass to charge ratio that we assign to this cluster. And you can think of this, these are somehow the features that we extract from an analysis, we call them ion peaks. The next step is then the annotation. Then we just look which common mass to charge ratio fits a certain metabolite based on the exact mass. So we go to the third digit after the comma. So this would be a 1 millidalton accuracy. And then we just say everything which fits here, we annotate to a metabolite. And in this window here, we have 4 common mass to charge ratios, but only one of them, this one indicated here, was annotated to a metabolite. And this metabolite is phenylalanine, an amino acid, and the mass matches actually quite good here. And we just do this over the full mass to charge ratio, and we use as a reference database, can use several databases. We can use the KEGG database, HMDB database, the human metabolome database. But what works good for us is that we use always the genome scale model of the bacterium or the organism that we are working with. So these are samples from E. coli. So what we do, we just annotate around 1,000 metabolites, which are in the current genome scale model of E. coli. And this is the model that is shown here. This is this IML 1515. This is the most updated genome scale model of E. coli. And all the metabolites listed there are then annotated to the mass peaks. That means we kind of only reduce the data structure. So it's very simple to the table that you've seen before. So the rows stay the same. We keep the same samples, but then we just keep those mass to charge ratios that match a certain metabolite and where we could actually annotate a peak as I explained before, with this peak prominence of 1,000 and the peak height of 1,000. And then we have this list of M to Z values that matches metabolite. And then we have some in-house software to explore these data. So here you see just the graph that you've seen on the previous slide for the window around phenylalanine. Then we have this big table here. You can scroll through that table, and these are all metabolites that are annotated to a certain ion peak. And that indicates you here in the pink line, you see the exact mass that you would expect for phenylalanine in the protonated form here. This is measured in positive mode. And then the dashed line, you see this common mass to charge ratio. And in this case, it fits actually very well with almost no difference in the mass. That's what you see in this row here. And then you can scroll through the table here. Just one other example. This is ATP. And here you see all the peaks of ATP in that sample of 250 strains. So the question is, how good is the method? And as I said, we made now kind of a stress test for that method, or we tested how good is it, how selective is it. And what we did is we used authentic standards, 160 authentic standards of E. coli metabolites, so mostly metabolites from primary metabolism. So we had them first, so we dissolved them, we bought them from Sigma, we dissolved them at a concentration of 1 millimolar, put them in 96 well plates, and then combined them with a metabolite extract from E. coli. So that at the end, we had this E. coli extract spiked with these individual samples at the final concentration of 1 micromolar. This is also more or less the concentration you would expect for, let's say, a abundant metabolite in E. coli in this extract. So we arrived at this 160 spike-in standards. You see here the categories. We had something from amino acid metabolism, nucleotide metabolism, central metabolism, so on. So it covered a diverse range of metabolites. And then we injected them, measured them with flow injection mass spectrometry. And then what we expected is that we just see the baseline or the signal from the endogenous metabolites indicated here in our sample. And then just a spike-in sample would show an increase of the respective metabolite. So this is shown a bit better here for nucleotide triphosphates. And you see actually that it works pretty well. So what you see here is again the mass to charge ratio, and then the signal intensity. And then we zoomed always into the ion peak of the respective metabolites. Let's take the example here of CTP. Here is CTP in the deprotonated form. This here is the expected mass, and you see this again here. And the dashed line, this is the common mass to charge ratio. And then you see in orange, this is the sample where we spiked CTP into the E. coli extract, and down here, the black lines, these are the 159 remaining spike-in samples. And then you can see we detect for all the nucleotides, both positive and negative mode, let's say this significant increase. And that's important for the next slide. I will just call this a significant feature, or simply a feature. These are ion peaks that are significantly changed, meaning they have a Z score higher than 3 compared to the other samples. So it's indicated here. So whenever we have a situation like this, it means it's a significant feature. And the first question is, you saw here in the previous slide, it works for all the nucleotides. So for which other of the 160 standards is it working so well? And you see here on the diagonal for almost all of our spike-in standards, I think for 134 spike-in standards, we see such a situation where we have a significant increase in the signal, either in positive mode or negative mode. You see this here. And how you can read these matrices here in the columns, you have the 160 spike-in standards, and then in the rows, these are the ion peaks of the deprotonated or the protonated metabolite. So let's look at this. Let's say in this row, we have the peak that was annotated, or that matches the mass of ATP. It's just an example that we've seen before. And then we'd see everything in gray would be the signals that we get here from the baseline from the E. coli extract, so nothing happens. Then only in one sample, we get this significant increase where the Z score is higher than 3. And this would be the sample here on the diagonal. And this is then the sample with the ATP standard spiked in. So we sorted the samples that this matches always on the diagonal. So based on that, we can say we get these significant increases for 134 out of 160 of our metabolite standards, but there's a big but. You see also here, there's some false positives, right? So these are significant changes of the ion peak of a metabolite which don't match the standard. So obviously something is going on, and we looked into this in more detail. So we looked now per spike-in standard, how much of these significant features do we detect for any ion peak, even one that is not annotated to a metabolite. And what you see here, these are 160 dots, and this is how much of these significant features, just some ion peak, which is significantly changing in that standard. And on average, we detect per spike-in standard 12 of these increases, right? So here, this box plot, you see that on average, we have 10 up to 20 significant features per spike-in standard, but there are also some extreme cases which have more than 100 significant increases of these ion peaks. And the most extreme is glycerol 3-phosphate standard, which has around, I think, almost 5,000 of these significant features. And this looks like this, that all over the chromatogram, here in green, this is the glycerol 3-phosphate standards. We have these increases in the ion peaks. So we were wondering, what is this? What is the origin of these features? And the first approach to explain these features for us was to estimate the mass differences between the features. So it's indicated here. So all possible mass differences. And then we just counted them. So we counted just overall spike-in standards. How often do we detect one of these mass differences? And they're shown here on, this is now a new M2Z vector. So it's the delta M2Z vector, and just how often we detect such a mass shift. And then if you look at the mass shifts that are very frequent, so let's say everything which occurs with a higher frequency than 500, you see that many of them can be explained, because all of the ones that we can explain are here with a little arrow, and you see what it is. Here, for example, in this case, this is the sodium adduction. You have the sodium adduct, and you can zoom a little bit in here. So we did this again with a very high mass accuracy of, I think, 3 millidalton. Then you see this peak that we get here. This is the sodium adduct. And then we just went through the literature and through databases to explain all possible or many of these mass shifts. And in total, we find that we can explain 52 of these mass shifts that we see in our spectra. And then we went on with these 52 mass shifts. We then tried to explain all of these significantly changed ion peaks that you see here. So this is the spike-in standard for diaminopimelic acid, DIAP. So you see in total, this is the protonated peak, but then we had these additional ion peaks. This one, so 1, 2, 3, 4, 5 additional ion peaks. And then by using these mass shifts that we see, we can then connect these mass shifts to the protonated form, and that way explain them. And there are different explanations. Not all of them make sense, but you see here, for example, just around that would be just the potassium adduct, the sodium adduct, and here's the 13C isotope peak that you get from this. This you would simply get by extending your reference list by the certain adducts. But then the other connections over the other nodes, which go over 2 edges, they are more difficult to explain than by extending the annotation list. And by doing this, what actually nice to see that already 1/3 of the features are explained by this. So you see again here, all of the 160 spike-in standards and the fraction of these additional features that we can explain just by using the mass shifts. So for some of them, like this example that you see on the left, we can explain all of them, but on average, we explained 1/3 of the mass shifts. Of course, these neutral losses or these mass shifts, this is the only thing that can occur, because it's also known that where in the electrospray we can have effects like in-source fragmentation. So here you see just the principle of electrospray. I think this was explained, I guess, in the previous lectures. But what can happen in the electrospray is that you have a fragmentation similar like you have it in a triple quad or in a quadrupole. So for example, you have a glucose phosphate molecule, and then just in the electrospray, you get the phosphate and the glucose molecule separated. This is one example where we saw it in our case. So this is the mass that matches to hypoxanthine. So hypoxanthine has this mass of 137.045. So this is the exact mass of hypoxanthine. And as expected, we see this in the hypoxanthine standard, but we also see it in the inosine standard and in the standard of IMP. And what happens is, here is the molecule. The total molecule is inosine monophosphate. Hypoxanthine is the molecule here indicated in blue. And what happens in the electrospray, we just get rid of the sugar phosphate, or we get rid of just the sugar in case of inosine, and then what is left is hypoxanthine. That's why for all of these three standards, we get a signal at hypoxanthine. So again, we tried to explain this systematically and then went to the human metabolome database to get the MS2 spectra of most of our standards. So we got 452 of them. We got the MS2 spectra, and then we just looked which of our significant features match a feature in the MS2 spectra. So what is the overlap there? And this is now here on the y-axis. You see for our standards, our spiking standards, which of these significant features do now match a feature in the MS2 spectra. And you see for many of them, we find multiple in-source fragments. And if we add them then to our network or to the explanation, we can actually increase the fraction of these significant changes of these ion peaks up to almost 50%. So a majority of these additional features that we detect are explained by neutral losses and by in-source fragmentations, and the rest is still unknown. So the last question is a bit, how problematic are these in-source effects? And to test this, we took again the genome scale model of E. coli, and then we ask how many of these of our features, so of the significant changed ion peaks in a spiking standard, are now annotated to the wrong metabolite, because in the best case scenario, only have one significant changes, and this is the ion peak that matches to your metabolite. But how many additional annotations do you get with that? And the good news is, in around almost half of our samples, we don't have interference, so none of these additional ion peaks actually match a mass in our reference list. This is good, but then for some of them we have one interference, so this would be one false positives, and then we have the peaks, probably those also with these many additional ion peaks, we have much more interferences, up to 35 actually. So in average, you can say that for one true positive, you have to expect one false positive in such a FIA screen. So I think we have 10 or 15 minutes left. So at the end, I would just show you a case study, what we can do now with this method. And what we do at the moment is we combine flow injection mass spectrometry with CRISPR, or in this case with CRISPR interference. This was one of our last papers where we developed a CRISPR interference library for E. coli. So we did for each gene that is involved in metabolism of E. coli, we made multiple knockdowns. So this is illustrated here. So in total, we had 1,515 genes. This is this IML 1515 model. And for each of these genes, we cloned 4 to 6 guide RNAs. And then we transformed them into E. coli, which had dCas9 on the genome. It was inducible with a PTET promoter. So you can really start in these strains from a wild type level at the inducer, and then the protein will slowly disappear due to growth. So in this study, we had a pooled CRISPR-I library. So they were all in one pot. And then we used next generation sequencing to see how they grow. We'll keep this short. So these are all these 7,177 strains. And the interesting thing was this cluster here on the right. It's just how abundant they are. We cultured them for 13 hours. And then you see everything without a growth phenotype here in these clusters. Nothing happens. And these clusters C and D, here the strains are getting depleted. So obviously they have a phenotype. And in cluster D, they have a stronger phenotype. And these are 253 genes. So there are more CRISPR-I strains because we have multiple guides per gene. And what we did then is we selected for each of these genes one strain and put them onto plates. So in total, these are 253 strains. They fit on 2.5 96 well plates. And what we saw so far, most of them work. We use proteomics to quantify if the protein is really downregulated, and you see here the statistics. So for the majority of these proteins, we have, let's say, strong decreases, more than a twofold decrease. And then in some of these strains, we could not detect the target, and only in 7 strains we show actually that there's an increase of the target. So most of them worked. And what we then did with these strains, we used them for flow injection, metabolomics. So we used made metabolite extracts from these cells. And here just see one example. So the data here is from all of these strains, and we zoom now into the to the mass of this metabolite here. This metabolite is lipid X. The exact mass would be again here at the pink line, and the dashed line you see the common M to Z vector. And what you see is in most of these strains it's impossible to detect it. So this is lipid X. It's actually a relatively polar metabolite, so we never see this in E. coli wild type cells. And also in the majority of the CRISPR-I strains, we cannot detect this metabolite. But what happens now, if we perturb the gene which is encoding the enzyme, which is using lipid X as a substrate here for membrane biosynthesis, we see that we get an increase in that CRISPR-I strain. So lipid X is increasing. That's what you see here. This is the red signal in this LPXB strain. It's also increasing if you perturb a little bit downstream. So LPXH is giving actually a stronger signal. So that's not clear why it's stronger. It could simply be because the knockdown of LPXH is just more effective. So this is still something we have to look into. But what is actually surprising is that if we perturb upstream of the pathway, LPXA, we also get an increase of a downstream metabolite. And this is a bit a paradox, I think, but we can explain it at the moment because we see that the cells are actually regulating against the pathway. So a knockdown can actually lead to an upregulation of the whole pathway. And that's why we might see this increase of lipid X. But it could also be, again, that we just have some fragments that occur from other metabolite. And then what I can also say is down here, these are also some other targets, these little increases here of genes that are involved in membrane biosynthesis. So we went now through all of the strains. So here is again the statistics of the proteome data, and here are statistics of the metabolome data. So in around 1/3 of these strains, we see a very specific increase, or these strong increases of the substrate. So this is nice. And in around 200 strains, or 80%, we see that some other metabolites are changing, which indicate that something is going on in these CRISPR-I strains. But I hope you agree that if we see such specific increases of the substrates, we can already infer from that it's very likely that the knockdown of this gene is successful. And then you can imagine it's much faster to measure the metabolome of these strains than the proteome of these strains to confirm that the knockdown is effective. So I think now I'm already at the example. So I think, Daniel, can you share the link?
Yeah, I think I pasted it already in the chat. Can you guys see it? If not, I can also repaste it. So it should be a tiny URL .com link with FIAMS 2022. And then, yeah, just click on it and it should pop up in your browser. And yeah, like this is.
Yeah. Exactly. And what we did here, this is another CRISPR-I strain. So it's one CRISPR-I strain and one control strain. And that control strain, nothing should happen. And in what you see in the CRISPR-I strain, we perturbed here an enzyme, which is in the middle of purine nucleotide biosynthesis, PUR-A. And if we knock this down, we see an increase again of the substrates, or the substrate effects, where inosine monophosphate is increasing. And then you should see some data like this. And I think you can just explore it now on the GMPS dashboard. I hope that works. And actually with that, I think we can do it like this, right? So while you guys are exploring this a bit, we can come to the end and the summary of the talk. And afterwards, it would be nice to have some discussion, to have some questions and answer, and simultaneously explore a bit the thing on the GMPS dashboard. So at the end, I think to summarize, I hope I could show you that with flow injection mass spectrometry, we can capture especially increases of many primary metabolites without the need for a specific LC column. So we can really capture polar, unpolar metabolites. But flow injection mass spectrometry has a low selectivity. You saw this if we use these single spiking standards, we get a lot of many additional significantly changed features. And as I said, so roughly we can estimate per one true positive, we get one false positive with these methods. So you always have to go on and validate what you see here, but still it's a great tool for large scale studies like the genetic screens that we're trying now at the moment, where we aim at thousands of samples in one screen. With this, I'm at the end. Just want to thank the people that did the study. So here in the middle, this is Niklas Farke and here's Torben Schramm. They worked mostly on the flow injection method and on the validation side. This is Ale and Andy here in the back. They did great work with arraying the CRISPR-I library, and also Ale, she extracted all these proteome and metabolome samples now out of 250 CRISPR-I strain. That was a lot of work and we were really excited about the data. So that was it. And with this, I'm at the end and I hope you have some questions.
Awesome. Thanks a lot, Hannes. Yeah, for like this nice presentation and introduction to VIA MS. Yeah, I think before we move on to the like more like informal discussion section, I just wanna yeah, thank all of you for like stopping by, and again Hannes for yeah, like giving the presentation today. Yeah, next time we won't have, so in 2 weeks we won't have the seminar because it's during like the metabolomics meeting in Valencia, where I see some of you. And then we will continue in July, where we have 2 remaining sessions before yeah, like the summer break, and then we may re-meet in the fall. And yeah, I just want to point those out because I think this is may now like leading like all up to like these particular 2 sessions. And that is like now we've seen like different methods such as like LC-MS data analysis with feature finding and molecular networking, but also like the flow injection analysis, what Hannes just showed. And what we end up there is typically that we get like a list of putative metabolites or features where we do not know what they are, but then what are we going to do with this data? So yeah, I think the next 2 sessions are going to focus on first the visualization of this data in Cytoscape, and then like some table juggling and multivariate and univariate stats of like that outputs. So yeah, without yeah, further ado, thanks a lot for stopping by, and hope to see you next time.